Now we're going to look at cell division. This is cells making more of themselves. This is one of the characteristics of life, is reproduction. There are two different types that we're going to look at here, mitosis and meiosis. We're going to start with mitosis. This is division of the nucleus in which chromosome number stays constant or the same. The goal here is to get exact copies. This involves body or somatic cells, and this is in humans specifically. As an overview, we're going to start out with a mother cell here. So within the nucleus, we have chromosomes that are represented. We have pairs, and these are going to be color-coded according to which parent they come from. So after DNA is replicated during interphase, we're going to have duplicated chromosomes, and after mitosis and these chromosomes divide, we're going to end up with genetically identical cells. So if you're comparing the chromosomes here and here in the two daughter cells to those in the mother cell, they're the same. What we want to note here is there's two kind of major phases in a cell cycle. We have interphase and then mitosis, which we're going to get into in more detail. During interphase, this is the phase between divisions, and even though you can't tell it from this diagram, most of the cell's life is spent here. During this time that the cell is not dividing, it obtains nutrients, synthesizes substances as needed, and in preparation for division, near the middle of interphase, DNA replicates. This is the S phase of interphase for synthesis. So during that phase, DNA doubles. The beginning of cell division is prophase. Your book further divides this into early prophase, prophase, and late prophase. The things to note about prophase is that it's the beginning of cell division. We have the nuclear membrane or nuclear envelope that disappears. We have spindle fibers that appear. We have spindle fibers appear that form between the centrioles as they move apart. We looked at centrioles earlier. Also to notice is that under a microscope you can start to see chromosomes. Chromosome itself means color body, chromo meaning color, and some meaning body, because when things were dyed under the microscope you could see these color bodies. So, quick review of prophase, this beginning phase of cell division. The nuclear membrane disappears, centrosomes migrate, spindle fibers appear. The chromatin that was within the nucleus condenses and chromosomes become visible. Each is composed of two sister chromatids. That means that the DNA has already replicated, so you have exact copies made. The centromeres attach to spindle fibers. The chromosomes have no particular orientation. We'll see in later phases how they do have a particular orientation. After prophase, we have metaphase, anaphase, and telophase. We're going to get into each of these in more detail. But note, after chromosomes had no particular orientation during prophase, during metaphase, they're all neatly lined up at this imaginary line called a metaphase plate. During anaphase, these sister chromatids are pulled apart, and during prophase, we start to see two distinct cells. During metaphase, meta meaning middle, the spindle fully forms and consists of poles, asters, and fibers. As we just mentioned, the chromosomes line up at the metaphase plate. During anaphase, the centromeres divide and sister chromatids are moved to opposite poles by fibers. The kinetochore spindle fibers shorten, pulling those daughter chromosomes to opposite ends of the cell. Polar spindle fibers push the poles apart. During this phase, the two sets of identical chromosomes become visible as each is moving to opposite sides of the cell. And the cytoplasm begins to divide as well. That's known as cytokinesis. And the cell membrane pinches inward. Telophase is the wrap-up. The nuclear membrane reforms, spindle disappears, and cytokinesis occurs. Cytokinesis, as we just said, is division of the cytoplasm. So started in anaphase, gets completed near the end of telophase. The chromosomes become more diffuse again, and the nucleolus appears in each daughter nucleus. Here, the migration of the two sets of chromosomes continues and is completed. The genetic material changes from rod-like chromosomes back to thread-like chromatin. That's what we mean by the chromosomes becoming more diffuse again. The spindle apparatus disappears, and we have two new cells called daughter cells that are formed. The acronym for mitosis is PMAT for prophase, metaphase, anaphase, and telophase. You won't be responsible in this class to know the differences between early prophase, prophase itself, and late prophase. But do know that during prophase, the nuclear membrane disappears. We have those spindle fibers that appear, seen here or here, and they form between the centrioles as they move apart. Metaphase, middle, and chromosomes are neatly lined up at the metaphase plate. Anaphase, sister chromatids being pulled apart. In telophase, we have two distinct cells. During meiosis, this is cell division in sex cells or gametes, particularly egg and sperm cells. This occurs in the life cycle of sexually reproducing organisms. Whereas mitosis was maintaining the chromosome, meiosis reduces the chromosome number by half. This provides offspring with a different combination of traits than that of either parent. So this is different from mitosis. Mitosis, we had genetically identical daughter cells, and meiosis, we have genetically different cells. Meiosis begins with one diploid parental cell, just like mitosis, but whereas mitosis had one cell division, meiosis has two. Whereas mitosis gave us two daughter cells, meiosis gives us four daughter cells. With mitosis, we had diploid cells. This means there are two copies of each chromosome. A haploid cell has half the diploid number, so there's a single copy of each chromosome. In meiosis, this involves pairs of chromosomes called homologs that are inherited from each parent. We'll see this information again as we get into genetics. So, during meiosis, this 2n equals 4 means that's a diploid cell, the 2n there. And 4 gives us the chromosome number. That's 1, 2, 3, and 4. So, the blue and the red represent a pair of chromosomes, one coming from the father, one from the mother. And so, same with these shorter chromosomes. After interphase, in which DNA is replicated, meiosis undergoes first one nuclear division and then a second. And so we're left with 4 haploid daughter cells. During the first nuclear division, we still have prophase, metaphase, anaphase, and telophase, but it looks a little different than in mitosis. During prophase 1, homologous chromosomes pair during synapsis. This is when they can exchange little pieces of DNA. During metaphase 1, we still have this metaphase plate, but rather than being in a single file line like we saw in mitosis, there's two. And so these are homologous chromosomes. One chromosome with the same type of information from the father and the other from the mother. What we see here with this kind of X-shaped pattern, that's a duplicated chromosome. So half of that, as we see in the mother cell, is a chromosome before duplication. In anaphase, we have homologous chromosomes that are separated and pulled to opposite poles. During telophase 1, daughter cells have one chromosome from each homologous pair. So we can see we already have genetically different cells here. Interkinesis is this in-between time between meiosis 1 and meiosis 2, which is the first division and the second division of the nucleus. During meiosis II, we have two daughter cells already, so this is showing the products from meiosis I. Meiosis II resembles mitosis a little more, but we already have genetically different cells. But prophase, we have chromosomes from each homologous pair that can be seen. Metaphase II, chromosomes align at the metaphase plate in a single file line. In anaphase II, the daughter chromosomes move towards the poles and are separated. Telophase II, spindle disappears, the nuclei form, and cytokinesis takes place. And again, cytokinesis is that division of the cytoplasm. So the result after meiosis I and meiosis II is that we have daughter cells that are haploid, and we have 4 of them. Comparing meiosis to mitosis, during prophase, in mitosis there's no synapsis. Synapsis and crossing over is when homologous chromosomes can interchange little sections of DNA. That's what's represented by these chromosomes here that have exchanged a little piece. During metaphase, we have the homologs that align independently in meiosis, but in mitosis, we see that single file line. Anaphase looks similar in that we have chromosomes being pulled to opposite poles, but during meiosis, we can see how genetically this is just a little different since we had the homologous pairs of chromosomes that lined up rather than individual chromosomes in mitosis. We can compare the final products of mitosis and meiosis. Compare the chromosomes here to those here. And we can see that these are diploid. There's a pair of chromosomes. And here, there's a single set of each type of chromosome for a haploid cell. Notice here that the daughter nuclei are genetically identical to the parental cell in mitosis, and the daughter nuclei are not genetically identical to the parental cell in meiosis. This table gives you further detailed information comparing meiosis I with mitosis. Pause this as a review.